Good morning, everyone. Welcome to lecture 18. So how was the exam? Alright, I see a few thumbs up, and I think one take-home message I got from talking to some of you before at the start of class is, when I throw a tantrum about a certain topic in class, pay attention, alright? So I hope it helps you to read me and also predict what the exam will be like based on what I lay the emphasis on during the lectures. Alright, so I even heard from someone that they listened to the lectures at like 1.5 times speed before the exam, so and it really helped, paid off. And so that's why we record the lectures and we make it available so that you can go back and review and see what I was emphasizing just before the exam. So I would highly recommend maybe using 3 or 4 days just to speed listen to all of the lectures before the actual exam. Alright. So today we are going to move on to the second part of the endocrine system. And after I get through what I have in mind, we will do some of the poll everywhere questions from the skeletal system. I didn't feel it was right to talk about those questions yesterday since the exam was yesterday, but today we'll wrap up those poll everywhere questions right from the skeletal system. So here's an overview of what we'll be discussing. I will start with the types of hormones which we mentioned yesterday, and I'll say what type of effects they can have further downstream. Can those hormones have either genomic or non-genomic effects? And what I mean by genomic is that is a hormone capable of affecting the transcription and translation process? In that case, it would be called genomic effects. Maybe it's affecting other organelles, then it would be a non-genomic effect. And then we talk about this concept of the types of stimuli for hormone release. So this bullet point here is only one slide, but I can't overemphasize the importance of this slide. This concept of what type of stimuli causes a hormone being released will be applicable for every single hormone we talk about. Alright, so that's an important concept. And having laid all of those general concepts down, we can move on to the specific endocrine glands. And today I want to focus on the hypothalamus and some functions of the pituitary gland, how they are controlled by the hypothalamus. So as usual, when we talk about a specific gland, please know its location, its structure, and also what hormones can be released by that gland, how the release of those hormones are regulated, like what is going to cause it to have an increased secretion or decrease the secretion. That's what we mean by regulation. Also, what are the functions of those hormones and what can go wrong if you have too much or too little of a given hormone. So keep in mind that hormones which are found in our body, they can be existing in two states, right? So notice that this purple circle here is indicating the hormone, and it is bound to this blue circle, which is actually the binding protein. And this cartoon is representing the sex hormone binding globule. So this is just a protein which is bound to the hormone. And notice that this is what is happening in the extracellular environment. But once that hormone enters the cell, notice that it can engage in either metabolizing an enzyme or it can bind to another hormone receptor. So the take-home message in this slide is that hormones can exist in two states. They can either exist freely in the cytoplasm, or they can also be bound to proteins. And we find that depending on whether they're bound or unbound, they can have a different sort of impact on the body. Alright, so that's the take-home message from this slide. So here's a question for you to consider. I told you that hormones can exist in either bound or unbound states. Tell me which of the hormones will be more functional for a longer time. Would it be the hormones which are bound to a protein, or those hormones which are freely in circulation? Let me know what you think, and then I'll explain what I think and why I think that way. Okay, so most of you said Bound hormones will be functional for a longer time, and that is correct, right? And then let me explain why. So this is a slide on vitamin D synthesis. That's what this picture shows. I'm sure you all will be able to explain what this is standing for, right? You see that when the UVB radiation from the sun comes in contact with the skin, it's going to form this 7-dehydrocholesterol, which is the precursor. And that will eventually be sent to the liver, where it's made into calcidiol, and that can in turn be converted into calcitriol in the kidneys, and then it acts on the small intestines. So we know how vitamin D synthesis is affecting multiple, or is influenced by multiple organ systems. But in this slide, I just want to draw your attention to this yellow box here. Notice that vitamin D is bound to a protein. Alright, once it is entering the bloodstream, whether it's vitamin D3 or D2, in order for it to be transported to the liver, it has to be bound to a DBP, which stands for vitamin D binding protein. So we find that if a hormone is free and not bound to a protein, it is going to be active only for less than an hour, right? And the reason it is active only for a short time is because there are multiple things which can inactivate the hormone. And let me talk about 3 of these things which can inactivate the hormone. Firstly, we see that if a hormone is not bound to a protein, it can very easily diffuse out of the bloodstream, and it can bind to receptors on target cells. And so we find that it's not going to be present in the bloodstream because it can easily diffuse out. Secondly, we find that hormones which are not bound to proteins, they can be easily absorbed and broken down by the liver or the kidneys. Alright, so that's why it has a shorter shelf life. And finally, we find that proteins which are not bound to hormones, those hormones will have a larger surface area where enzymes can bind and break them down. Alright, and that's the reason why the hormones which are unbound have a shorter half-life, right? They will be degraded very quickly. So we find that thyroid and steroid hormones both will be bound to proteins, and so they will be functional for a longer period of time. So now we're going to switch gears and talk about how hormones can have an impact on a given cell based on whether they can enter in or whether they're going to use secondary messengers. So we find that these G protein coupled receptors, they are going to be involved when a hormone is unable to enter into the cell. So what do you think would be the property of such a hormone which cannot get past the lipid bilayer? It would be hydrophilic, right? That's why it can't get past the lipid bilayer. And so in such a case, that hydrophilic hormone is going to bind to a receptor on the cell surface, and it's going to use an intermediary to bring about its effects. Okay? And the intermediary which we are referring to here would be an action which is regulated by the G protein coupled receptors. So this G protein coupled receptor is referring to a complex of proteins which are going to require GTP, right? That's the reason why it's called G protein coupled receptors. And the hormone which binds to this receptor on the cell surface will be the first messenger. I told you that the endocrine system is all about communication between different parts of the body and cells. And so when a hormone binds to a protein receptor, that the hormone is the first messenger. And this binding is going to activate the inactive G protein complex. And so once this G protein has been activated, it is going to trigger the release of secondary messengers which will eventually carry out its function. And there are a couple of secondary messengers we'll talk about, and one of the secondary messengers is cyclic AMP. So one of those secondary messengers is cyclic AMP. So let's see how cyclic AMP is going to be produced. So keep in mind that when this G protein has been activated, it will in turn activate an enzyme called adenylate cyclase. All right, and it's going to cause ATP, adenosine triphosphate, to be converted into cyclic AMP. All right, so what does cyclic AMP look like? So here is a structure of the ATP molecule. Notice that there's the adenosine group linked to the ribose sugar, which is in turn connected to 3 phosphate groups. So it's adenosine triphosphate. Now, cyclic AMP stands for cyclic adenosine monophosphate. So it only has one phosphate. So the remaining 2 phosphates, they're going to be removed as pyrophosphate and it's released. And then there's going to be a cyclic group formed between the remaining 1 phosphate and this ribose sugar. Alright, so that is the reason why it's called cyclic adenosine monophosphate. Will I be holding you responsible for these structures? No. But I do hope that you can appreciate why it's called cyclic AMP versus its precursor ATP. And notice that this cyclic AMP is going to activate kinases, right? So a kinase is an enzyme which is capable of adding a phosphate group. And once that kinase is activated, it can have other downstream effects. It can cause the opening of ion channels. It can activate other enzymes as well. So let me just briefly tell you something about opening of ion channels. Right? When you move on to the nervous system, we find that everything is going to be based on the movement of ions. Right? We will see how whether it's the signals which your eyes receive from electromagnetic waves, or if it's mechanical signals which reach your eardrums, everything will be converted into the movement of ions, and we'll find that some G-protein coupled receptors are involved in the functioning of the nervous system as well. So this is all going to integrate. But for now, just keep in mind that because this hormone can't get into the cell, it's going to bind to a receptor on the surface, and as a result, the secondary messenger is cyclic AMP. So examples of hormones which use this type of mechanism is epinephrine, norepinephrine, calcitonin, which we'll talk about, parathyroid hormone, ADH, ACTH, FSH, LH, and TSH. So all of these hormones will make sense to you. These names will make sense to you by the end of next week. All right. So we find that sometimes the binding of a hormone to a G protein coupled receptor can also convert cyclic AMP into AMP. And notice that unlike cyclic AMP, which will cause the activation of certain enzymes, AMP is going to have the opposite effect. It's going to reduce the enzyme activity. So notice that in some cases the G protein activation can decrease the cyclic AMP effect in the cytoplasm. And examples of where the cyclic AMP activity will be decreased is by the action of epinephrine and norepinephrine. Keep in mind that I told you that in the previous slide, epinephrine and norepinephrine can increase cyclic AMP activity, and here I'm saying it's decreasing it. Right? So it seems like an apparent contradiction, but it really depends on what type of receptor the epinephrine and norepinephrine are binding. Right? If in some cases the receptor may cause an eventual activation of cyclic AMP, in other cases it can have an inhibitory effect. All right, so it really depends on what the receptor is. So the enzyme which is responsible for converting cyclic AMP into AMP is the phosphodiesterase enzyme shown right here. All right, and if you have a pen and you have your slides marked, I would highly recommend you put an important sign on this, right? You can expect a question on this topic. Because this is pretty foundational to other concepts which we'll be discussing in the upcoming lectures as well. So we see we talked about cyclic AMP as being one type of secondary messenger. Now let's move on to talk about other examples of secondary messengers. Alright? And so I just want to draw your attention to this figure here. Notice that calcium has been mentioned like 3 times in this slide. Alright? And so the time when I mentioned that calcium is a secondary messenger is when we talked about smooth endoplasmic reticulum. right? In that slide I mentioned, it can store calcium, and calcium can act as a secondary messenger. Here's where I'm fulfilling what I said previously of how calcium can be a secondary messenger. So notice that in this case, hormone is going to bind with a protein receptor. So the hormone is the first messenger. And here also you have the G protein being activated. And notice that there is this phospholipase C which is activated, right, as a result of this G protein being activated. And phospholipase C can act on the channels which can permit calcium to come into the cell. So that is one thing which happens. You also see that this phospholipase C can cause the activation of this DAG, which stands for diacylglycerol, and also IP3, which stands for inositol triphosphate. Right? So it's going to convert phospholipids into these compounds. And what this IP3 does is it's going to move into the cytoplasm, and it will act on the reserves where calcium is stored. Right? And can you tell me which organelle calcium is going to be stored in? I just now mentioned it. Smooth endoplasmic reticulum. Right? So it can act on the smooth endoplasmic reticulum, and also this SER is that smooth endoplasmic reticulum, and that calcium can have further downstream effects. So in one case we see that it can activate PKC, which stands for protein kinase C. The term kinase means it's going to add a phosphate group. C stands for it needs calcium in order for that enzyme to be active. Right? So you see how calcium is activating enzymes further downstream. And also the calcium can bind to calmodulin, which is a protein. We'll discuss about this protein again in the smooth muscles, and that can cause more enzymes to be activated further downstream. So the examples of where calcium can be a secondary messenger is in the case of epinephrine, norepinephrine, and oxytocin. So keep in mind that epinephrine and norepinephrine are going to use secondary messengers, and the secondary messenger can be either increasing the amount of cyclic AMP or decreasing the amount of cyclic AMP. That happens by two different mechanisms. Two different enzymes are involved: adenylate cyclase versus phosphodiesterase. And finally, also calcium can be used as a secondary messenger by epinephrine or epinephrine. All right, and let me just give you a quick preview into what epinephrine and norepinephrine does. When you have the fight or flight response being activated, let's say the bear jumps in front of you, that's where epinephrine and norepinephrine will play a role, and that's going to activate secondary messengers. All right, are we clear on this, folks? This is a super important concept. All right. Okay, so moving on, let's see how hormones can have either genomic effects or it can have non-genomic effects. And so this cartoon here is showing a steroid hormone, okay? And these steroid hormones can affect the rate of DNA transcription which takes place in the nucleus. Alright, so transcription is where, based on the DNA sequence, you have mRNA being produced, and that can be affected by certain hormones. So notice here that once the transcription or translation is affected, it can have a downstream effect on what proteins can be synthesized, and depending on the proteins being synthesized, it can affect the function of that specific cell. So keep in mind that this hormone is able to diffuse right through the diffuse right into the cytoplasm because it is hydrophobic. So there is no barriers preventing it from entering into the cytoplasm. And once it enters into the cytoplasm, I told you that it has to be bound to a specific protein. And in this case, notice that the protein to which it binds is actually a nuclear receptor. And this means this protein can carry this hormone straight into the nucleus through the nuclear pore as shown here. And once it's inside, that nuclear protein and the hormone complex can bind to the DNA, and that can trigger the transcription process where you have the RNA being produced. Right? And so once the mRNA is produced, it can be moved back into the cytoplasm where proteins can be synthesized, and the protein can even be sent out of the cell to act on various target tissue. Right? So is this clear how you have genomic effects which are possible for steroid hormones, and how the fact that steroid hormones are hydrophobic allows them to have a genomic impact? Is that clear? All right. So we find that there can also be non-genomic effects, and we will consider how the thyroid hormones are capable of doing this. Right. So this cartoon here depicts the thyroid hormone. It's got these two benzene ring structures, and we find that thyroid hormones will be capable of going into the nucleus, as I mentioned in the previous slides, and regulating the transcription of specific genes. Or it can also bind to a receptor which takes it into the mitochondria. Can you tell me why we discussed mitochondria in the previous unit? What is important for metabolism, for making ATP, right? So we see that the thyroid hormone can increase the rate of ATP synthesis, right? So we find that it can have non-genomic effects as well. So is this clear, folks, how steroid hormones and also hormones which are hydrophobic can have genomic and non-genomic effects? So here's a question which, do you have any questions before I move ahead? Okay, if not, I wanted to show you this question which was found on RPA 1, but I was notified by a student, this doesn't make sense, and I was like, yeah, you're right. This should be in RPA 4, right? RPA 3, which is the endocrine system. So can you read this question and tell me what options could be right? So let me read this to you. So protein contains a trans activation domain, a DNA binding domain, a nuclear binding domain, and a hormone binding domain. Right? So just that first sentence, does it seem to relate to what we just now discussed? If a protein has got a hormone binding domain, it's got a place where it can bind the DNA. What can you think? What can you say about such a protein? So do you think it is likely true that it was synthesized on the rough ER, or do you think it can contain a signal peptide? Do you think it could be glycosylated, or do you think it could be synthesized on free hormones? Right? So you folks, you can turn to your neighbors and discuss, and we can just walk through each of these options. This is a really fantastic question, which is integrating information from what we discussed in Unit 1, with transcription, translation, and ultimately it's tying into the endocrine system function. Okay, folks, so let's talk about this, right? Do you think a protein which bound to the steroid hormone could be described as such a protein? Yes, okay. Let's talk about option 1. Do you think it could be synthesized on the rough endoplasmic reticulum? Okay, what is the major property of the steroid hormone? Hydrophobic, right? It's hydrophobic. So, what is the property of the signal peptide? It's going to be hydrophobic, right? So, the signal peptide is a sequence of hydrophobic sequences which will be recognized by the SRP, right? Signal recognition peptide, And what will happen to that specific protein? It will be taken into the rough ER, right? So that means option 1 should be true, A is true, and also option B will be true. Is that clear, folks? Why option A and option B are likely true about this protein, which has a region where it can bind the hormone, where it can bind the DNA, and all of that. And once it is in the endoplasmic reticulum, right, it can undergo some post-translational modification, right? And do you think glycosylation is a potential post-translational modification which takes place? Because even if it doesn't happen, the glycosylation doesn't happen in the ER, it's going to move from the ER to the Golgi apparatus where glycosylation can take place, alright? And the one option which is not likely true is that it's going to be synthesized on the free ribosomes, because if option A is true, that it is synthesized on the rough endoplasmic reticulum, option D can't be true as well. Is this clear, folks? So this is a question which is integrating multiple concepts, and I hope it makes sense when we discuss the endocrine system. Right? Any questions about? Oh, and as a side note, I just put the slide in this morning, so I'll upload the updated slides later today. Alright. So moving on, let's talk about how the hormone action can be either upregulated or downregulated. Right? So what we mean by downregulation is that the synthesis of a specific hormone is going to be reduced, right? You're turning down the volume. Or upregulating is when you have more of a hormone being produced, you're turning up the volume. So this panel on the right hand side, let me walk you through this, right? So this is just showing the hypothalamus, right? And how eventually, as a series of reactions take place, you have GC, which stands for glucocorticoids, being released from the adrenal cortex. So what you need to know is that this pointy arrow means that it's activation, right? Whereas if there's a minus at the end of the arrow, it means inhibition. So we see that something from the hypothalamus is causing an activation in the pituitary gland, which is in turn causing an activation in the adrenal cortex, and as a result we have glucocorticoids being released. And so notice that these glucocorticoids can also go back and have an inhibitory effect on the pituitary gland, or it can have an inhibitory effect on the hypothalamus, as denoted by the minus sign at the end of the lines. Are we clear as far as that's concerned? So if you have a pointy arrow, it means it's increasing the production. Minus arrow, it's inhibiting it, right? So this is a fundamental concept we need to understand, because we have positive feedback and negative feedback, which we will consider in the endocrine system. So what we're going to answer is, what can cause downregulation and what can cause upregulation of, let's say, glucocorticoids in this example. So we find that if there's going to be a decrease in the number of hormone receptors, right, that can cause downregulation. So keep in mind that these receptors on cells are all blank proteins. We talked about these proteins in lecture 2 or maybe lecture 3. They are the transmembrane proteins. Alright, so these receptors having an increase or decrease in the number is all going to be dependent on whether you have more protein synthesis, right? These receptors can be easily incorporated with the cell membrane. So if we have more number of receptors, you can have an upregulation. If you have fewer receptors, it's going to downregulate the synthesis of a hormone. We also find that if a cell is becoming less sensitive to a specific type of hormone, that can also cause a downregulation. So if you have fewer receptors, cell is less sensitive, it's going to downregulate that hormone synthesis. So let's go to the other side of the coin. What can cause an upregulation? Right? So if there's going to be an increase in the number of receptors which are available, that can cause more of that hormone to be produced. And similarly, if the cell is going to be having a greater sensitivity to the hormone, you have more of it being produced. Alright? So again, the take-home message from this slide is, what does the pointy arrow mean? What does the minus arrow mean? Why is hormones being upregulated, or why is it being downregulated? And just keep in mind that once glucocorticoids are released, they can have a number of effects as shown here, and we'll be talking about some of these effects in the upcoming lectures. Okay, so now we go on to a very important concept which is in one slide, but it will be applied throughout all the hormones we discussed, right? So hormone release can be triggered by three things, right? It could be triggered by a humoral stimuli, or it could be triggered by a hormonal stimuli, and this could be triggered by a neural stimuli, right? I like this slide a lot because it's very easy to write exam questions, alright? And so let's talk about what is the difference between humoral, hormonal, and neural stimuli. So humoral stimuli is something where there is a change in the concentration of certain components in the blood, right? So if you have a change in blood glucose level, you have a change in blood calcium level, you have a change in the phosphates in the blood, all of that is going to be humoral stimuli. And the word humoral is referring to the fluids in the body, and so it makes sense that humoral stimuli is referring to changes in the concentration of certain components in the blood. Hormonal, on the other hand, is where you have either a hormone coming in, binding to a specific receptor, and as a result of that, you have other hormones being synthesized. That's hormonal. Or maybe you don't have sufficient hormones coming in binding, and that's why you have the activation of other hormones being released. And finally, neural stimuli is where you have neurotransmitters, which are binding as a result of signals coming from the neurons, and that's why hormones are released. So let's walk through a few of these, a few examples for each of these, right? Let's start with humoral stimuli, and the example here is the blood calcium levels will be monitored by the thyroid gland and also the parathyroid gland. So if you have a decrease in blood calcium levels, you have the parathyroid hormone, which will be released by the parathyroid gland, and if you have too much calcium, the thyroid gland will release calcitonin, and as a result, it can decrease calcium levels. So this is just a sneak preview to how calcium levels are regulated. We will discuss this in greater detail in the upcoming lectures. Moving on to hormonal stimulus, notice that the hypothalamus is going to secrete a number of hormones, right? And these hormones can in turn act on the posterior lobe of the pituitary gland, or they can also act on the anterior lobe of the pituitary gland. And so the hormones which are produced by the posterior and anterior lobe, they can act on specific targets like the adrenal cortex, they can act on the thyroid gland, they can act on the gonads, right? So we find that this is an example of hormonal stimuli. So keep in mind that the hypothalamus releases hormones which will go bind to the pituitary gland, and the pituitary gland is going to release other hormones which will bind to target organs. So is this clear how hormonal stimuli is brought about? Yes? Yes, okay, great. Neural stimuli is where you have signals sent from the nervous system. So the spinal cord is part of the central nervous system, and we find that there will be these preganglionic sympathetic fibers. What we mean by preganglionic, I explained when we discussed the nervous system. But keep in mind that the spinal cord is sending signals through the nerves to the adrenal medulla. So this is the inner part of the adrenal gland, and as a result, it's going to cause the release of epinephrine and norepinephrine. Right? So we've talked about epinephrine and norepinephrine, I think, in at least 4 or 5 slides up till now, and now we know how it is actually being activated. When you have a flight or fight response because you see something scary, that will eventually cause epinephrine or norepinephrine to be released through neural stimuli. Okay, any questions at all, folks? Move on to start our discussion with the pituitary gland and how it is controlled by the hypothalamus. So notice that the pituitary gland is shown right here. It's got three parts. It has this anterior lobe shown in pink, and then there is this posterior lobe shown in yellow, and in the middle you have this intermediary region known as the pars intermedia. And the reason I point out these three parts of the pituitary gland is that each has a unique function and they can release different types of hormones. And this is going to be very important, and so we see how the pituitary gland is protected by this bony encasement. So it's this is bone called the sella turcica, in which the pituitary gland is protected. And this pituitary gland is connected to the hypothalamus by a narrow stalk known as the infundibulum. And keep in mind that this anterior lobe is capable of secreting 7 hormones, right? So it's going to produce and secrete 7 hormones, whereas the posterior lobe is only releasing 2 hormones. So what is the difference between just releasing versus secreting and producing, right? And the analogy which I get is that if I were to buy a cake and just distribute it, right, I didn't make it, so I'm just releasing the cake to everybody. But if I bake it and then I distribute it, then I'm doing this production and secretion. So similarly, keep in mind that the hormones which are released from the posterior lobe, they're actually made at a different location, and the location is the hypothalamus. And so there are neurons which extend from the hypothalamus down to the posterior lobe, and that's why they release the hormones. So now let's take a closer look at this pituitary gland. So notice that the anterior lobe of the pituitary gland is called adenohypophysis, and it's going to have these darkly staining nuclei, right? Whereas the posterior lobe is having a very light staining. So the anterior lobe is made up of glandular epithelium, and the nuclei will stain pretty darkly, whereas the posterior lobe is made up of nervous tissue, and the nervous tissue has got these myelin sheaths, which is mainly made up of fat, and that's why it doesn't stain as well, and it's going to stain darkly. And overall, we have 9 important hormones which are released from both these regions. So 7 from the anterior region and 2 from the posterior region. And all of these are capable of binding to extracellular receptors, and as a result of binding to the extracellular receptors, they will release cyclic AMP as the secondary messenger. And both the anterior region and the posterior region, the release of hormones from there, it will be controlled by the hypothalamus. Right? So let's see how the hypothalamus can control what the pituitary gland can release. So the hypothalamus has got 3 major functions. Right? Firstly, we see that it's going to produce 2 hormones, the antidiuretic and the oxytocin hormone, by the neurons in the hypothalamus, and they will release it in the posterior lobe, right? And that's how these 2 hormones are going to be released from the posterior lobe. Secondly, we have the regulatory hormones, which are also released by the hypothalamus, and they can go down to this anterior lobe of the pituitary gland, where they will cause more hormones to be released. And finally, the hypothalamus has also got these nerve fibers which eventually reach the adrenal medulla. And in the previous slide, I showed you how these nerve fibers, they go from the spinal cord. So keep in mind that they go from the hypothalamus down to the spinal cord, and from there they enter into the adrenal medulla. And that's how you have epinephrine and norepinephrine being released as well. So you know that endocrine hormones have to be transported in the blood, right? So the question is, where is the blood vessels through which these hormones are transported? And the answer is, here it is, right? So there's a system known as the hypophyseal portal system, through which these hormones will be carried. And notice that there are two arteries I want to point out, right, which are bringing blood to this hypophyseal portal system. So the term hypo means below, right, and physeal is standing for the stalk, for the word stalk. And you may wonder, where is the stalk? And the stalk is this infundibulum which connects the hypothalamus to the pituitary gland. So the blood vessels are going to go in this infundibulum. And the word portal is just referring to the fact that there are blood capillaries. So where does the blood come from for these capillaries? So there is this superior hypophyseal artery which is going to deliver blood to this hypophyseal portal system, and the blood from here is going down to the anterior lobe of the pituitary gland. Right? And so we find that all of the hormones which are released by the hypothalamus, they move through this superior hypophyseal artery down to this anterior pituitary gland. Right? And whatever hormones are released from this anterior pituitary gland, they can travel out through these veins. Notice that there is also a second artery called the inferior hypophyseal artery that is bringing blood to the posterior lobe of the pituitary gland. And the hormones which are released from the posterior lobe, they will also be carried through this vein down to the rest of the body. So notice that the blood flow is going to be unidirectional, right? So it's not going to backtrack, blood is going in one direction. So any hormones which are released by the anterior and posterior lobe, they'll have to circulate through the rest of the body before they can come back to the hypothalamus to have a negative feedback effect. That's possible because the blood flow is unidirectional. So let's talk about one hormone which is released by the hypothalamus, right? And we find that there are two hormones which are released. We have both ADH and oxytocin, and I want to focus on oxytocin. So the paraventricular nucleus and the supraoptic nucleus are both found in the hypothalamus. Both are capable of releasing ADH and oxytocin. And keep in mind that ADH and oxytocin, they both have a very similar structure. Both are made up of 9 amino acids, and only they differ in 2 of those amino acids, right? So both can be produced by either paraventricular or the supraoptic nucleus. But it's found that the supraoptic nucleus produces ADH to a slightly higher extent, and the paraventricular nucleus produces oxytocin to a slightly higher extent. So both of these hormones will travel down through the infundibulum, and eventually they can be released by the posterior lobe of the pituitary gland. So let's talk about the oxytocin function. So the word tosia means childbirth, and one of the major functions of oxytocin is to help in childbirth. And how it does that? It's going to cause the contraction of the uterus and also dilation of the cervix. And as the pregnancy progresses, the receptors for oxytocin will keep increasing in the uterine canal, and they're going to reach a peak of receptor production just before childbirth. And so we see the smooth muscles are most sensitive to oxytocin during just before childbirth. And we find that the one hormone that seems to have a positive feedback effect, which we'll discuss, is oxytocin, right? And we find that as the fetus moves through the uterine canal and it's going to press against the cervix, it will cause a positive feedback to be sent to allow more oxytocin to be released. So the more it contracts, the more it's going to, the fetus presses against the uterine walls, and more oxytocin is released, and there will be an increasing intensity of contractions. So this is the first function. The second function of oxytocin is to help in milk ejection, right? To let down the milk. Now keep in mind that there is a difference between milk let down and milk production, right? So when an infant starts suckling on the nipples, we see that the production of milk is due to prolactin. And prolactin is a hormone produced by the anterior lobe of the pituitary gland, not the posterior lobe. We'll talk about prolactin and the when we come back tomorrow. But also, as the infant suckles, there's going to be neural stimulation which will be sent to the hypothalamus, right? And from there we have the production of oxytocin, and the oxytocin is going to move down into the posterior lobe of the pituitary gland. And oxytocin is going to act on the myoepithelial cells. So these are type of smooth muscle cells which will cause these lactiferous ducts where the milk was produced to start squeezing, right? And as it squeezes, it's going to cause the milk to be pooling into the lactiferous sinuses, and eventually it will be discharged through the nipples. So keep in mind that the production of milk is through prolactin, but the eventual letdown is through oxytocin acting on the myoepithelial cells, which causes the milk to be squeezed out. So as the infant starts tasting the milk and suckles more and more, they have this positive feedback effect where more oxytocin is released, and so there will be a greater letdown of milk. Is this clear, folks, how oxytocin does two things: milk letdown and also in the delivery of the baby. A third function of oxytocin is that it can also help in bonding between the newborn and the parent. It's not only the bonding between mother and child, but also the father and child as well. And oxytocin has a reputation for being a love hormone, so it can help closeness and also plays a role in the sexual response as well. Right. So with that, let me summarize what we discussed till this point. So we talked about the genomic and the non-genomic impacts of hormones. We find that hormones can either act on the DNA, that's the genomic effect, or it can act on other organelles in the cell. And we see the hypothalamus, how it can control the posterior lobe of the pituitary gland to release oxytocin. We didn't talk about the other hormones, but keep in mind that the hypothalamus is also going to have an impact on the anterior lobe as well. And so when we come back tomorrow, we'll talk about all of these other hormones which are being released by the anterior lobe, and also we'll talk about the function of ADH as well. So with that, we'll end a couple of minutes early, and see you all back tomorrow.